Afternoon, and for those of you joining us from across the country and around the world, good morning and good evening. Welcome to our webinar on hollow fiber model research. I am Amy Liebman, President of NTM Info and Research, and I will be serving as moderator for today's session of Science from Your Sofa. On behalf of all of us at NTMIR, thank you for joining us. We are delighted to welcome Dr. Jacco van Ingen and Jelmer Raimakers, PhD candidate, as today's featured guest speakers who will take us inside their lab as they teach us about hollow fiber model research and how it's being used to shape research in the development of therapies for NTM lung disease. Jacco van Ingen, MD, PhD, is a consultant clinical microbiologist and head of the Mycobacteriology Reference Lab at Radboud University Medical Center in Nijmegen, the Netherlands. He has authored over 200 papers on mycobacterial disease in peer-reviewed scientific journals. He's the co-author of recently published international guidelines on NTM treatment, and he has consulted on diagnosis and treatment of NTM disease all over the world. His research group focuses on the development of new treatment regimens for non-tuberculous mycobacterial disease and studying how mycobacteria deal with antibiotics and the immune system. Jelmer Raimakers is a PhD candidate at the Department of Medical Microbiology, also at the Radboud University Medical Center. His main focus is finding new therapies against non-tuberculous mycobacteria using antibiotics that can be inhaled. In hollow fiber infection models, he exposes NTM to antibiotic concentrations attained in the human body to evaluate the efficacy of treatment modalities. During today's webinar, you can ask questions using the Q&A function on Zoom. Click on the Q&A icon on your screen, type your question into the box, and then click send. At the conclusion of the presentations, I will read the questions from the queue for Drs. van Ingen and Raimakers to answer. Today's webinar broadcast is being recorded and will be posted online at a later date. So if you missed the answer to a question that is asked, you will have the opportunity to hear it again. Our next webinar topic will be Let Go of NTM, Investigational Use of Nitric Oxide as an Antimicrobial Agent in Refractory NTM Lung Disease, with guest speaker Dr. Rachel Thompson on Friday, May 7th, 2021, 9 a.m. Australian Eastern Standard Time. And yes, we will post this information with other time zones as well. And on June 13th, our mental wellness series with Dr. Devin Smith of National Jewish Health kicks off with the first webinar, Sleep 101, Sleep Basics and Solutions for Improvement at 1 p.m. Eastern Daylight Time. You can learn more about NTM Info and Research and sign up for our electronic news at ntminfo.org. And you can also find us on Instagram, Facebook, Twitter, YouTube, and LinkedIn. And now I would like to turn this event over to our two guest speakers so they can take us inside the lab and explain to us what hollow fiber research is and how it's being used.
Wow. Well, thanks a lot, Amy, for a world-class, introduction of the two of us and our work. So welcome to all of you. We're terribly excited to be here live with you from our laboratory in the Netherlands. So my name is Jacco van Ingen. And we'll be your sort of hosts tonight. At least it's nighttime for us. And we'll take you through our lab, focusing on what we do, and that is finding new treatment regimens for NTM, especially NTM lung disease, through various preclinical studies. So what does preclinical mean? Well, it means we do clinical trials without having to harm any animal, and especially without having to harm any patient. So some of you may already be enrolled in a clinical trial, because we're in a very fortunate era where there are new treatment modalities for NTM being tried in the context of clinical trials. But if you want to join a clinical trial, the one thing you want to know is, okay, but if I'm going to get like an experimental treatment, how good is that? And, you know, is there good science behind this new experimental treatment? Is it safe? Is it likely to be effective? Um, and that's part of what we do. We make sure that only antibiotics or other treatment modalities that are absolutely effective, and in fact, hopefully or presumably more effective than currently recommended treatments, make it into clinical trials. So that is what we do. That's our goal in life, to make sure that only the best antibiotics make it into clinical trials. So you never have to worry if you join a clinical trial or hear about one, that you never have to worry about, you know, what is in there. So we will switch to a small series of PowerPoint slides. Of course, that's always, you know, the big ooh moment. Is this going to work out right? So we're going to share our screens. So sorry, Amy, we're stopping yours.
Okay, good. Please.
Yes. So there we are. So Jelmer and I thought, you know, the topic of today should be positive, not just hollow fiber research, which is fascinating in its own right, but what we need is more effective antibiotic treatment of NTM disease, especially NTM lung disease. So here at Radboud University Medical Center in Nijmegen, the Netherlands, tiny country, the size of Maryland, in Western Europe, um, we are a reference clinic for NTM, pulmonary disease. So patients from throughout our small country come to us for diagnosis and treatment, especially when, you know, first-line treatments have not been successful or have been difficult to tolerate. We're also a reference laboratory, meaning that we perform drug susceptibility testing, for example, for essentially the entire country and for some laboratories in our surrounding countries. And of course, you know, as a medical doctor involved in treatment of NTM pulmonary disease, you know, it takes you like a little over a week to realize that, you know, our current treatment regimens are just not good enough. They're, you know, they're toxic. You need to use them long term. And that does not come with a brilliant outcome. So, you know, despite using 3, 4, sometimes 5 antibiotics for, you know, a year, 1.5 year, 2 years on end, you know, there's not a guarantee of a cure. And to us, you know, that's something that we just can't tolerate. You know, we need to do better. So we need to do clinical trials of new or existing drugs that might help improve treatment outcome. But then you need to select the right drugs for those trials first. And that's what we do. So we have set up what we call a pipeline. It's an awful word, but it's used for a lot of things. So it's a series of connected technical works, a series of techniques that we use to assess how good is this antibiotic really. Um, and usually that starts off with relatively simple testing. How high a concentration of this drug do I need to kill Mycobacterium avium or abscesses? And that's called a minimum inhibitory concentration, or an MIC. And that tells you a little bit, how active is this drug really? Do I need a tiny amount to kill the bacterium or quite a bit? In the end, that might also influence how high doses might need to be in a clinical trial. So the next thing we do is we look, is this a drug that's active in itself, or does it also boost the activity of other drugs? Or the other way around, can other drugs boost the activity of our new candidate drug? So are the two synergistic? Um, Once we've assessed that, we are going to look more in depth into how does this drug really kill the bacterium? Um, and then you start looking into things like, is the killing faster or more extensive if I increase the dose of the drug? Or doesn't that really matter? Is it more a matter of just exposing the bacterium to the drug for a long time, and then it will have an effect, almost regardless of how much? That type of information is very important to later on think about, is this a drug that we're going to give a low dose 3 times a day or a high dose once a day? What is best to really kill these bacteria and still be safe and tolerable for the person taking the drug? The next step is literally the next step, because then we're going to make it a little more human-like, because the NTM, in fact, all mycobacteria survive in our body with several strategies, but a very important one is that essentially they lock themselves up inside the cells of our immune system, the macrophages. And by locking themselves up there, they also create an additional barrier for antibiotics, because the antibiotic has to come into the cell and inside the cell come to the bacterium to be able to kill it. So whenever we work on a potential new drug, what we test is, does this drug get to the inside of our immune cells, macrophages in particular? So does it really get to where the bacteria are? Because you can imagine, if it doesn't really do that, it's probably not going to be a very good drug. And I think one of the recent successful examples of new drugs being developed for NTM pulmonary disease, so the ALIS or the liposomal formulation of amikacin, it was amikacin put into liposomes, so little spheres of a cholesterol-like fat, only to get inside those cells. So just showing you how important that feature is. You need a drug that can get to the bug. So that we try in again, these killing models, but inside macrophages. And then if the drug has passed all those tests and we're almost ready to sign off on saying, okay, this looks good, then we do the ultimate test. And the ultimate test is exactly what Jelmer Rijmakers will inform you about, because he is our expert on that. And that is using the hollow fiber pharmacodynamic model. Usually, back in the old days, I would say, if a drug was almost ready for a clinical trial, it would be put into an animal model, mouse, guinea pig, and for some, primates. That's something that, from an ethical perspective, we do not like, but also for NTM, these animal models are really complex, because a mouse simply is not a human, and to make NTM infect a mouse, you have to kill parts of the immune system of the mice. So they're no longer animals with a normal immune system. And the disease that you then get from the NTM in these mice is also more of the disseminated disease that you get in humans. that are severely immunocompromised, that have weakened immune systems. So I'm never too fond of those animal models because we don't really know whether they can predict the outcome of a treatment for NTM pulmonary disease, because most of these mouse models don't develop a serious pulmonary disease. They develop a serious disseminated disease in essentially all organs, very different from NTM pulmonary disease. So to get closer to NTM pulmonary disease, we use the hollow fiber model. But that, of course, is a model in a lab. So you have to make that as human lung-like as you can. And that is what Jelmer will explain you about. So here are the initial graphics. So on the left of the screen, you'll see the actual hollow fiber model setup. And as you see, it's a big, well, it looks like a fridge, but it's actually warm on the inside. It's a stove. On the right, that's where all the mini lungs are. And on the left side of the picture, you see the bottles with the red liquid and all the pumps and wires. Jelmer will explain you exactly what all that is. And the picture in the middle actually shows Jelmer performing a bronchoalveolar lavage inside our fake lung. So it's what we have recreated that looks like the interior of a human lung. And in there, by doing this lavage, Jelmer can determine, okay, how many mycobacteria are still alive despite the antibiotic treatment that we're giving them. And he can also test the concentrations of the drug that we're working on to see, okay, are we achieving the exact concentrations over time as they are in the human lung? So are we really modeling the human situation good enough? And how effective is it? So that's visually what that looks like. But of course, the model itself is a little more complex. So I'm very, very happy to have the brighter person on board. Jelmer will briefly guide you through the hollow fiber model and how it simulates the human body and drug treatment. So Jelmer, please.
Hello, good evening. Here it is evening. And when people get diagnosed with NTM pulmonary disease, they most often will get antibiotics to treat these infections. And they most often come within a pill. And when you take this pill, it immediately starts to dissolve in the mouth and in the stomach in smaller particles. So this is already a preparation for the uptake of the antibiotic inside the human body. When it enters from the stomach into the guts, it gets taken up into the general circulation, and we can mimic that using our hollow fiber infection model, using the drug syringes in the green box in the bottom left. By pumping drugs into our system at a general at a speed determined to really mimic the uptake of the antibiotics inside the lungs, we can really mimic the exact concentrations and the rate in which the human body takes up these antibiotics. And when the antibiotics are taken up into the general circulation, it spreads all across the body. And in the middle, you can see the green box with a flask. And this is actually our general circulation because the drug syringes pump the antibiotics into our general circulation. And from this general circulation, it expands to all of the body, also, for example, the head, but most important is the lung because that is actually where the infection takes place. So from the general circulation, we pump it through these hollow fiber cartridges, we call them. And this is basically a fake lung. And in this fake lung, we have also added a cell that looks a bit like a macrophage, because NTMs, these bacteria go indeed inside macrophages. So by adding these macrophage-like cells into our lungs, we really can mimic how the life for the bacteria is inside the human body and inside the lung. And by flushing through these cartridges, we can actually draw samples from them and determine how many bacteria are still inside that lung, but also how much antibiotic is indeed in that lung, because the antibiotic in the general circulation can be a bit different for certain antibiotics than is in the lung. And when we have simulated the lung concentrations of the antibiotics, we also need to model an exit strategy for the antibiotics, because when you take an antibiotics, these will not stay inside your body for your entire life. After a while, these antibiotics will be filtered out of the circulation by your kidneys. But for some antibiotics, they first need to be broken down into smaller particles by the liver. And from the liver, they get back into the general circulation again, and then they get filtered out by the kidneys. So that eventually all of the antibiotics are also flushed out of your body. So that is this bottom part. On the bottom left, you can see these brown bins. And from the general circulation that I just showed you, we can pump out the antibiotics just like the human body would do eventually. So within this, within our model, we can, with the drug syringes, we can really simulate very well how the human body takes up any drugs from the guts, and then how they spread throughout the whole body. And then also more specifically inside the lung, because that is where the infection really happens, and also in these macrophages. And after we have mimicked that, we also mimic the filtering from the body and the cleansing from the antibiotic.
Yeah, so we had the plan. I mean, this is a tough one to digest. I still feel like that every time, even though we work with the model, it's still tough. So to digest this, we thought, why not just try and walk into the lab? Here we are. Welcome to the lab. So you're currently inside our pharmacodynamics lab. Jelmer has already put on his lab coat, and he will give you a tour of two experiments that are currently ongoing. One time kill kinetics experiment, and of course, the hollow fiber model. So the first thing Jelmer will show is a time kill kinetics experiment in which we are looking at a classic inhaled drug called pentamidine and see, does that work against mycobacteria? So Jelmer will show you what that looks like. So as you see in these bottles, you see a liquid culture medium, but it's completely cloudy. And it's cloudy because it is full of mycobacteria. And in this case, that's Mycobacterium abscessus that has really grown very well. Now, that doesn't worry us too much because that was a bottle that had no antibiotic. Here you see a similar bottle, again, with a liquid, but if you look closely, you'll see the liquid medium is really rather clear, which means that there are very, very few bacteria alive. And these bacteria have been exposed to high concentrations of this weird antibiotic called pentamidine. So in very high concentrations, it has an effect on Mycobacterium abscessus. And we look at this over time to see, okay, how strong is the effect? Does it last over time? And if you increase the concentration or the dose, does that make it more effective or not? So that's the time kill kinetics. And you see the remainder here shaking at the bottom. And we shake them to give them the best options to reach their nutrients in the broth that they're in. So next step is the hollow fiber model. And I have to take a few steps back because it is really rather large. So this is the entire hollow fiber model. And as we explained in the slides, this model really goes from taking the antibiotic, it reaching your gut, coming into the bloodstream, and then into the lungs. And that's what we model here. So Jelmer will guide you through. So what you see here are pumps that have the antibiotic. So essentially, this is the patient's meal, and the antibiotics are in there. And then they go from the big syringe. They go through these hoses, which essentially are the gut, into the hollow fiber model proper, which is the simulated patient. And if Jelmer opens it, you'll notice the sound, which literally is the heartbeat of the system. So I guess you're hearing the chugga, 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 which is literally the heartbeat of the system. And the heart and circulation are reflected by the bottles that you see with the red liquid. So from these bottles, there's another hose there going into this tube with the red caps on both ends, and those are the lungs. And what Jelmer is now doing is using these syringes to mix sort of the content or the liquid inside these lungs, which is the equivalent of our simulated patient undergoing a bronchoalveolar lavage. And from these syringes, we can take this fluid and count how many bacteria are still alive at this point in therapy. And this is what Jelmer has been. So this is the way in which we try to simulate clinical trials without having to bother animals. So no need for animal models, and without having to expose patients to drugs that maybe do not work. So with this very, very complex model, we really hope that we can find the best drugs to then go into clinical trials so that no patient in a clinical trial for NTM pulmonary disease will ever be treated with a drug that in analysis phase we say, oh, it didn't work, or oh, it did well, less than we had expected. So that's what we do, selecting the best drugs to go into clinical trials.
Definitely.
So this model from today will run for a total of 3 weeks to see, okay, how effective is this potential new drug against Mycobacterium avium complex pulmonary disease. So that was the tour of the lab. Well, Jelmer will update you on our most recent findings, and I think, please, the one Jelmer will detail you on is quite spectacular. But drugs that we have previously worked on and that proved really to do really well in these models are clofazimine, for which we've now also completed a randomized controlled trial to compare a clofazimine, ethambutol, and azithromycin regimen to the standard rifampicin, ethambutol, azithromycin regimen for Mycobacterium avium complex, or MAC. pulmonary disease. So clofazimine is one of the drugs that we've been working on for a long time. And more recently, we've also worked on tedizolid. And we've done some really exciting work on this new drug called SPR 720, which we're very happy has also progressed into actual clinical trials. So that drug also got our seal of approval, like, okay, if this drug is safe, then we say it's also good and it's an effective drug. So we're very happy that that moved into clinical trial phase. Um, and So we've done, as I said, some minor work on tedizolid, which is also a very new drug, which has been used in NTM treatment a little bit now, but it's also a drug that we found to be pretty active. We've worked on bedaquiline, which sadly we found not to be really effective. So in order to use that, you need to boost its activity. So that's ongoing work. But what we're particularly excited about is our some of our more recent work, and Jelmer will get you up to speed on that.
So if I just told you from the gut, antibiotics can be taken into the general circulation and it spreads all across the body. But what I didn't tell you is that it does not spread evenly across the body because antibiotics in the lung sometimes have a higher concentration than in the blood plasma. So here you can see if we mimic these antibiotic concentrations inside the blood or inside the lung, we have followed how many bacteria are still alive within our hollow fiber infection model. If we look at the red line in the blood, we can see that it gradually does increase. So the higher the red line, the more bacteria are inside our lung. And if we look at our lung, at the lung graph, we can also see that there is indeed growth inside our within this regimen, but it is less growth than if we mimic the blood concentrations of antibiotics. So that is actually a good thing for us because this means that our antibiotic regimen that we use, if we simulate it in the lung, it does quite a lot better than inside the blood. Our model shows this because the bacteria that we measured inside the blood, if we mimic those concentrations, those antibiotic concentrations, then the bacteria tend to grow faster inside the blood than inside the lung if we mimic the same standard regimen with azithromycin, rifampicin, and ethambutol. So this therapy would do better indeed with lung infections than with disseminated infections. So good question. So we have also investigated minocycline as a trial inside the hollow fiber infection model. And as you can see, instead of the standard rifampicin, ethambutol, and azithromycin regimen, we have replaced rifampicin with minocycline in this regimen. And as you can see on the right, again, the higher the lines, the more bacteria are inside our lung compartment. As you can see, there is much more killing within our minocycline therapy because the line actually goes down. So that means that we actually kill the Mycobacterium avium, sorry. But after 7 days, we do see some regrowth. But if we compare the green line with the red line, we can see that it does quite a lot better than our standard therapy if we simulate this within our hollow fiber infection model. So based on these results, we can also tell that minocycline is a little bit better than the standard therapy. And therefore, it is also of interest that we test minocycline in a clinical trial to really prove that it is also more effective in patients. So we started a PK study, which is a study um, that is focused on determining the antibiotic concentrations inside the blood and inside the lung. And if we know these concentrations, we can also simulate this back into our hollow fiber infection model. But we also know that we reach a high enough concentrations inside the patients. Maybe we should dose a little bit higher, or we should dose a little bit lower, or maybe we should completely change the dosing regimen by maybe dosing multiple times a day or even less times a day, which would be better because it's easier to take one pill a day than 3 or 4, for example. So what we want to focus on in the future is how to tame a Mycobacterium abscessus, because Mycobacterium abscessus is quite a resistant mycobacterium, and it isn't really susceptible to a lot of antibiotics. So we really want to focus on how can we treat these mycobacterium really effectively, and also. Yeah, that's also a good point, because Mycobacterium abscessus also tends to kill cells. And within our hollow fiber infection model, these Mycobacterium abscessus also tends to kill our macrophages. So we're finding out how can we ensure that this Mycobacterium abscessus doesn't kill our macrophages, uh, and to bring our treatment regimen to a success. And for both McAvium complex and for Mycobacterium abscessus, we are going to focus on inhaled antibiotics, because if you got a lung infection, it would be a good thing if you would inhale the antibiotic, that it would be directly on the site of action, that it can directly enter the macrophages and access the mycobacteria. And examples thereof are liposomal amikation, which Jacco told is the Alice, which also really looks really promising. Pentamidine, which is actually a really old antimicrobial drug, but we want to repurpose this antimicrobial drug for the treatment of McAvium complex and M. abscessus. Last but not least, also tigecycline, which is also an inhaled antibiotic, and we want to try to treat Mycobacterium abscessus with this antibiotic. Now, at this moment, clofazimine-based regimens are being tested in clinical trials, so that is really something to look forward to for us. And for McAvium complex, we also want to test a 2-drug regimen, which is only ethambutol and azithromycin, versus the 3-drug regimen, which is the rifampicin plus the ethambutol plus the azithromycin. And definitely because rifampicin causes some toxicity within our human bodies, we want to test if we can get rid of that drug and just be as effective with 2 drugs as 3 drugs. A colleague of mine is really focusing on what NTM are doing. So if we are going to administer drugs to a patient and they get to the NTM, what do these NTM actually when they encounter these antibiotics on a genomic level? For example, do they mutate so that they get resistant to these antibiotics, or do they create um, other proteins, so as a mechanism to counter these antibiotics? So really, what do these NTM other than other antibiotics, so that they do live and other bacteria often die, because NTM are quite resistant to many drugs. So that is also where we in the future will definitely focus on. So We would like to thank our medical microbiology team, of course, but also our hmm? Yes, the picture of a colleague of ours is gone, unfortunately. And we also would like to thank our colleagues from the pulmonary disease department and the pharmacology, which really focuses on the antibiotics, of course, and also our colleagues from the infectious diseases department and our microbacteriology teams. Thank you.
Thank you both very much. This was very informative. We have a number of questions um, that from we have a number of questions from the guests who have joined us for today's webinar. So I think we'll just dive right into those first questions. So let me see. Let's get the first one. Somebody actually sent this one right in. They want to know, does this regimen make sense with the minocycline? Does it make sense with minocycline when resistance appears in the standard of care therapy? And sorry, can you sit right in front of the microphone, right in front of the computer when you answer so we can hear?
Yeah, so sorry, because of the COVID measures, we have to sort of stay 1.5 meters apart. Yeah, so that's a tough one. Is minocycline a potential treatment option after other regimens have failed? Well, at this point in time, what we see looks more like that minocycline has a role in first-line treatment. So replacing rifampicin in the first treatment regimen that you would start for a patient with NTM pulmonary disease. We have not studied it yet in the setting of adding it to a regimen, if that regimen has not proven successful in, for example, the first 6 months of treatment, like what was done in the trials of the inhaled liposomal amikacin, or ALICE. That was added to a regimen that to that point had failed. That's not how we currently look at minocycline. We see minocycline as a potential, we need to do the clinical trial, but as a potential replacement of rifampicin with less interactions with other drugs, hopefully less side effects, because minocycline is a drug we know well, particularly from dermatology, where many people have used it long-term for the suppression of a skin disease called acne vulgaris. So we're quite comfortable with giving the drug for a long time. We also give it to patients with the fish tank granuloma. It's an NTM skin disease by Mycobacterium marinum. People affected by that disease also get minocycline treatment for 6 months on end. And toxicity, fortunately, is not a big deal. So we see for now, it might have a role in first-line treatment.
Okay, thank you. So we have several questions. We have someone who's asking 2 questions. The first one is, are you also extending your research to diseases that include gram-negative infections like Klebsiella, Enterobacter, Stenotrophomonas, which are common in NTM patients, and particularly those with resistant infections?
With our model, we are able to indeed simulate these kind of infections, but within our research team, we will continue to focus solely on NTM infections, NTM lung infections, in the at least in the near future.
And are you using your hollow fiber model to study omadacycline, which, as I understand, is something that's starting to be looked at more and more for sepsis treatment?
Well, not at this moment, but Jaco, will we do that in the future?
Yeah, that's still a topic of ongoing discussion. we have been in contact with the company that produces this drug. In fact, we have occasionally used this drug for Mycobacterium abscessus treatment here in the Netherlands. It's kind of hard to get by here. We have to have it flown in from the US, but it's something that we're definitely interested in. Also, because it's an oral drug, so it's something that I think we're already quite accustomed to working with this type of drug. So we would love to. That's the answer. We would love to, because we do think there's promise for this drug. And so if the company wants us to do it, we'll do it. We cannot access, we have no access to the drug ourselves. So we can't just order it and just go ahead, but we're definitely interested in working on it.
Okay, we have some questions now relating to clinical trial design and some regulatory issues. Can you summarize some of the main differences and the pros and cons of hollow fiber model research using NTM versus using NTM animal models? So for example, talking about the differences in pharmacokinetics, toxicology and safety testing, tissue distribution, and so forth.
Yeah, so safety testing is something that we do not do inside our lab. We really just mimic the human pharmacokinetics. So that is also a pro of the hollow fiber infection model, because animals do have different pharmacokinetics, which is the concentrations that are followed inside the human body or in animal models. So the concentrations reached in animals and in humans, they differ a bit. And with our hollow fiber infection model, we can really mimic the pharmacokinetics inside the humans without the use of animal models or directly administering just to patients. We can really do a really well preclinical work with concentrations that will be reached inside humans. So those are at least pros for the hollow fiber infection models. And it's also from an ethical point of view, it's better to use non-animal models compared to animal models, because you will infect living animals. They will get hurt, and it's really stressful for these animals. So it's also from an ethical point of view, definitely better to use a hollow fiber infection model.
And can either of you speak to the reactions of the regulatory agencies for investigational new drug submissions, their reaction to hollow fiber data?
Yeah, so a few years ago, the FDA already positioned the hollow fiber model as a suitable screening tool. So a hollow fiber model data are sufficient to allow you to have your drug progress into phase 1 or phase 2A clinical trials, so early stage clinical trials. Similar advice has been put forward by the European Medicines Agency, provided that you know or have a very good estimate of the drug concentrations as they are over the day in the human body, and that you have simulated just that. So if your simulation is human and good, then you can use it to have a drug progress into a clinical trial. So it has been cleared by the FDA for that purpose. You always have to realize, and you may have noticed that in Yelmer's data as well, that you see a drug regimen killing. So the number of live bacteria goes down and then goes up again. Now that suggests treatment failure, drug resistance, but you have to realize hollow fiber models have no functional immune system. So what normally your immune system would contribute to, attacking the NTM does not happen in the hollow fiber model. So the hollow fiber model really looks at the impact of the antibiotics, separate from the potential impact of your immune system. So that is, from a scientific standpoint, a pro, because you get a very clean view of what's going on. It's, of course, also a con, because ideally you'd like to see what the drug adds to the human immune response, and that's not what you get from this model. But mind you, that's also not what you get from animal models, because then you get what a usually damaged mouse immune system does to NTM, and that's also not what we see happening in humans. So both the animal models and the hollow fiber model have that con, that it's, you don't see the effects that the human immune system has.
Okay. So we have a question about, I guess, some of the regimens, and this, I guess, goes to the ability of you to use the hollow fiber models to combine various drug regimens. But they're asking about, I guess they're asking about clofazimine. And so have you tested clofazimine with any of the other drug regimens?
So yeah, we indeed have tried to test clofazimine within our hollow fiber infection model, but unfortunately, clofazimine tends to bind to plastic a lot. So when we pump clofazimine into our system, it binds to the plastic tubing that we use. So therefore, it's for us, we are unable on this moment to really simulate these antibiotic concentrations used reached inside the human body. But we will continue to try this in our model, and to also combine it with other drugs, such as ethambutol or azithromycin.
Okay.
We
Question.
have modeled working with clofazimine in collaboration with our friends in, I'm pointing towards Dallas, Texas. Of course, that you figured that one out. No, so we've tried that in collaboration with our friends in Dallas, Texas. Again, as Jelmer explained, having lots of difficulties with this weird drug, but we did get very good results in terms of efficacy when we modeled the use of clofazimine, and that also sparked the clinical trial that we've been doing here in Nijmegen over the last few years, which we now finalized, and which, sneak preview, I can tell you, clofazimine, ethambutol, azithromycin performed exactly similar, exactly identical to rifampicin, ethambutol, azithromycin in the clinical trial. Just don't tell anyone. But yeah, so we're working on clofazimine, but it's a tough drug to work with. It's a weird molecule. But well, we're trying to find ways to work with it in the hollow fiber model, also to see maybe, if you change other parts of the regimen, can we make it more effective? And we're also doing a clinical study here at the moment, looking at what is the best dose of clofazimine, because it's a quaint drug, and how it distributes through the human body is quaint. So we're investigating different dosing regimens at the moment in actual in our patients, to see which dose gets to the best blood levels in as short a time as possible.
Okay, thank you. We have a quick question about the clinical trials, which I think I can probably answer, but somebody wants to know how we can learn about the actual clinical trials. So NTM Info puts information out when companies give us information about the clinical trials. We put it on our website, we send it out via email, we put it out on social media. There's also clinicaltrials.gov, which I know is a little bit difficult for people to use, and I know they are currently going through a redesign process using focus groups to determine how best to redesign it so that it's easier for the public to use. So those are two very good avenues to learn about clinical trials, and I suspect pretty much every other country, including the Netherlands, has a similar portal for pulling information about clinical trials. That's sort of centralized to the government because it's overseen by an agency and regulated. Next question relates to, I guess, a publication from last year. It's about M. kansasii. Can it be treated without the use of rifampicin?
Absolutely. Yeah. Sometimes we have to. Not everyone tolerates rifampicin all that well. So occasionally we have to treat patients with regimens that do not contain rifampicin. So again, our colleagues in Dallas, Texas, have done a fair bit of work on Mycobacterium kansasii using the hollow fiber model, quite similar to what we do. And there too, they find very promising data on other antibiotics that could really have a role in treatment of Mycobacterium kansasii. So, for example, a drug they are very interested in is moxifloxacin, a fluoroquinolone antibiotic, also used in drug-resistant TB. And that, for example, also shows very potent activity. So, yeah, treatment without rifampicin is definitely an option. At this point in time, I would not recommend it. But if someone can simply not tolerate rifampicin, yes, there are options.
And would these kinds of hollow fiber models work to study regimens for other strains as well, like Xenopi?
So we can actually choose the strain we want to use, so we can test all types of NTM bacteria.
Okay.
Provided that they kind of keep the cells alive. That's what we struggle with Mycobacterium abscessus, because some strains of abscessus they really harm our cells, like they probably do to our macrophages in real life. So sometimes you then need to look for a strain of that species that's a little tamed. So it's a little less virulent. And then if you can make it survive in the cells, yes, you can put it in the hollow fiber model and study treatment. Mycobacterium xenopi is definitely on my list. It's a big deal here in the Netherlands. We see a fair amount of patients. That's a tough one to treat. So yeah, that's definitely high on our list to work with.
Is there one particular area in the Netherlands where there's a higher concentration of NTM, or is it just that it's all over?
Our country is the size of a mid-sized American town, so it's all over. Our country is really too small to say anything of that. And yet, in our small country, we do see that some species tend to occur around specific cities or in areas. Like in the United States, Mycobacterium simiae, another really tough one. If it causes disease, it's very tough to treat. I know in the US, for example, it's frequently found in parts of Texas, like around San Antonio. In the Netherlands, it's found in The Hague, where our government resides. So in that city, there's a lot of simiae. In the rest of the country, it's essentially absent. For other species, we also see regional difference in their frequency of their isolation.
You had talked previously about studying bedaquiline, and you'd said your findings were that it was not as effective with MAC. Is that correct?
True.
Okay.
Yeah, and also our experiences in using it to treat our patients have not been good. I'll be very frank about that. They've not been good. We've even seen resistance developing despite treatment with regimens with multiple antibiotics that were active, and then including bedaquiline, we would still see resistance to bedaquiline emerging in the MAC bacteria that were causing disease. But it's always important to realize that, this is a last resort drug. So we typically tried it in patients who had failed on multiple rounds of treatment. So whether it was like not a good drug or just terribly bad disease is very hard to make out from this. But yeah, overall, it's not as good for MAC as it is for TB. I think that's something we can safely say.
And are you guys studying other kinds of treatments that are not antibiotics, or are you focused specifically on antibiotics with hollow fiber?
Yeah, so the hollow fiber model is really made for antibiotic treatment. But I must say, we are also studying phage therapy. Technically, mostly we don't treat NTM patients with phages, but we are looking into phage therapy and, how we can use phages or maybe phage antibiotic combinations, looking for potential synergy. Yeah, so phage therapy is something that we work on. It is not our core business, to say the least. It's no one funds that type of work, so it's it's something that we do on the side.
Okay. So with we know a lot of patients who have NTM infections tend to get reinfected at some point in the future. There's a high rate of reinfection. Are you looking at treatments with an eye towards something that has a lower toxicity level and might also help with like the suppression of recurrence of infections?
Yeah, we do, but I think Jelmer really worded it really well. So the toxicity part is not something that we can monitor from the microbiology lab. So there's dedicated other researchers working on, how, why these drugs are toxic, how that toxicity works, where do your side effects really come from. The prospect of studying drugs that can help prevent disease recurrence is incredibly important. It's something that I would love to study. The hollow fiber model, in the way we use it, is not really set up to do just that. But I have told many people who were, working on clinical trials and setting up clinical trials with new agents, please in the future also do a study of using your agent after regular treatment to prevent recurrences, because indeed these recurrences drive me mad. It's just, why? How can these bacteria just sort of pretend they're gone and then be back? So is that a new infection? If so, how can we prevent that? Or is it just that they're still inside the lungs, but they're sort of in hiding? And then, 12 months later, there they are again. So that's something that we are investigating at this moment. So in our lab, we use whole genome sequencing to identify mycobacteria. So we can also provide a DNA fingerprint of every isolate. So if after treatment there's another positive culture, we're like, no, it's Mycobacterium avium again. And we can provide a DNA fingerprint to see, is it the original strain, that we treated you for 18 months for, to see, is this recurrence of the old infection, or did you just, run into a new strain? So that's something that we are investigating as part of our routine diagnostics. But how to prevent those recurrences, that's really tough. And I don't think the hollow fiber model is very suitable for that purpose. I'm looking at Jelmer, who's also, nah. Now, the hollow fiber model is really looking at killing is this regimen going to kill the NTM or not? But preventing the recurrence is hard. In TB, there are some animal models that kind of look into this. That has not been developed for NTM, as far as I'm aware. So at this point, that's something that we still only need to tackle by clinical trials.
Do you have any information on the side effects of minocycline, especially compared to the side effects that we know occur with rifampin and rifampicin, the rifamycin drugs?
Well, minocycline, of course, is a tetracycline antibiotic, which means that there's a couple of well-known side effects. One of them is, that uneasy feeling, a bit nausea. That's something that occurs with essentially all tetracycline antibiotics. The other famous one is hypersensitivity to sun. So if you go sunbathing while using minocycline, you can get this nasty, itchy rash. So sunbathing is kind of not allowed when you're using minocycline or essentially any other tetracycline antibiotic like doxycycline, or if you're being treated for Mycobacterium abscessus, tigecycline. So those two, the rash upon sunlight and nausea, are the two most common ones. Rarely, minocycline can induce a lupus-like autoinflammatory syndrome. That's very rare, but that's the one big worry that anyone prescribing minocycline will always have. Like, okay, I need to monitor for this lupus-like autoinflammatory syndrome. It's really, really rare, but that is something that you're always thinking of and watchful for.
So somebody's asking about your research. They want to know where can they find copies of the research you've published, particularly on clofazimine. And I guess the follow-up to that is, can you send us some of the more recent published research, and maybe we can put some of it out for our patients to look at as well?
Yeah, so we both we strongly believe in open access publishing. So for us, it's incredibly important that what we publish in scientific journals is available to anyone. So most of our scientific work is. So the hollow fiber model work on clofazimine, we have sadly not been able to publish yet because we were not confident that it was really good enough. So that's why we're still working on that. But the prior work on clofazimine is published. So yeah, so all the ones that are published open access, we can easily share. And well, technically for things that are not open access, we would need to ask the journals that published it for allowing us to distribute it. But for the clofazimine work, I think it's no problem. We can easily distribute that. And all the other things, well, you can use PubMed as a source. It's an online sort of library where you can look through scientific literature. And there you can find our work, and you can easily see which ones are available by open access. So you can just download and read it. We're very happy to send them to you, Amy, for potential distribution.
Oh, that would be great. I'm sure our patients would really enjoy reading some of that, and so would we. Somebody has a question about
might cure your insomnia too.
Yeah.
Just putting it out there.
Don't be so sure. We're very curious people. We like reading this stuff. So we have a question about inhaled medications, the liposomal versus other formats of antibiotics. Do you think liposomal format formulations of other antibiotics would be less damaging
Less damaging due
or
to
less
direct.
difficult to tolerate, maybe? But certainly in
um,
terms of side effects, might it be, easier to tolerate and less difficult for the patient to handle?
Yeah, for some antibiotics, it might be, because if the antibiotic itself gets stuck to the inside of the throat, it might give some coughing reflex. And if it's packed inside a liposome, so this cholesterol-like fatty acid, then it might prevent from this coughing reflex. Yeah.
And somebody's asking about genetic engineering. They want to know if genetic engineering plays a role in improving the efficacy of antibiotics. I'm not sure. It doesn't clarify genetic engineering, like of what. Maybe I, I'm not sure. I'm not sure what they're talking about engineering. But
Well, so what's our other colleague? Well, you should have her on next time. She's also absolutely brilliant, like Jelmer. So Jody Schildkraut, our colleague, works on genetic engineering of NTM, but
oh,
we use that to study how they tolerate our antibiotics, because we've studied several mechanisms that particularly Mycobacterium abscessus employs to defend itself against our antibiotics. So what we've then done is just take them out of their DNA. So we have genetically engineered Mycobacterium abscessus to kind of lose part of its immune system against antibiotics, to prove that then it indeed becomes more susceptible. So we show that, yes, this mechanism is crucial for it to defend against antibiotics. So that kind of genetic engineering of the NTM we do as part of our scientific experimentation um, to see how do these bacteria work. Yeah, so that's what we do in terms of genetic engineering.
that sounds like a whole separate webinar.
It is.
So somebody was asking about the sputum tests, and they're asking, how did the sputum type tests measure the actual amount of the bacteria in the lung? And they were asking for a little bit of clarification about how that is simulated in the lab with the hollow fiber.
Yeah, that's a good one. So you're absolutely right. You don't walk into your doctor's office and, he said, well, last month you had 15,000 Mycobacterium avium bacteria in every milliliter of sputum that you coughed up. And yet we do measure how many mycobacteria are in sputum. Um, you don't always see those numbers, but in clinical trials, for example, we do. And in routine diagnostics, we don't count the actual number of live mycobacteria, but we do look at some proxies for that. Like, how long does it take for the culture to become positive? If there's a lot of bacteria to start with, that will just be 3 or 4 days for MAC. If there's very few MAC bacteria, it might take up to 14 days, 20 days. So we have a bit of a measure how many bacteria are still alive inside the lung. Is that something that we use, that information in the hollow fiber model? Absolutely. Because the amount of mycobacteria that we bring into our lung in the hollow fiber model equals the mean of what we see in our patients at the start of treatment. So the number of bacteria in the lung in the hollow fiber model is roughly the mean of what we see at the start of treatment in our patients. And this is actually a hot topic at the moment, looking at how many bacteria are there, because we're slowly realizing that that also maybe determines in part your chance of achieving a cure with antibiotic treatment. If there's a lot of mycobacteria in the lung, it is much harder to attain cure or to become culture negative over time during treatment than it is when the amount of bacteria at the start of treatment already is very low. So it's definitely a topic that has a lot of interest in the scientific community at the moment, for particularly for NTM.
Okay. Thank you. You have managed to answer all of the questions in the queue. So thank you very much. Thank you so much for taking, oh, wait. I spoke too soon. Oh, somebody wanted to say thank you and great work. We had a number of people chiming in to say thank you for the work you're doing. It's fascinating, and we're very excited at how helpful it's going to be for us. So thank you so much. And thank you for taking the time to take us through your lab. And yeah, we really appreciate everything that you're doing for us. Thank you for presenting to us today. And we look forward to you presenting results of more of your work probably next year sometime.
Yes, please. Well, it was a great pleasure. As always, Amy, it's always a great pleasure. And I really hope that next year there will also be a conference again.
It'll be
Because
nice to
those,
see everybody in person.
yeah, that's usually the highlight of my year, coming to your conference and meeting some really cool people that have driven me to my hotel because, you're not taking the bus, brother. And, I have tons of crazy stories of, coming to your conference and having a fantastic time. So I hope that
Well,
Jan and
great.
I can join you live next
We
year.
really
And
hope to have an in-person conference next year. We really do. So we get to
yeah,
see everybody again. We miss everybody. Thank you everyone for joining us, and we look forward to seeing you next month at our next webinar. And everybody stay safe, have a wonderful week, and we will see you soon.